quiz in here during the last 10 minutes of the period. It's going to be on basically this chapter that we're on now, which is the mitosis chapter. Though we're about to finish the mitosis chapter, and we're going to start in on Mendelian genetics next. All right, and both next week's laboratory activities are Mendelian genetics, so it'll consume us for the next week or so. But the last part of this lecture on mitosis was all about cancer. And I guess the major concept can be summarized as this, is that cells in your body can undergo mitosis where they divide to make more cells, but that's a very tightly controlled process. The body has to make sure that it uses these growth factors to tell the cells when they're supposed to divide. And even when the cells are supposed to divide, there's other things that control their divisions. There's this thing called contact inhibition, where if the cells are dividing but they start running up against other neighboring cells, they know they've divided enough, and so they stop dividing. And there are proteins inside the cells called tumor suppressor genes that are there to sort of act as brakes for the mitosis process. But when all those mechanisms break down due to mutations, the cell starts dividing in an uncontrolled way. That is what a cancerous cell is. A cancer cell is. And just to review some terms here, proto-oncogenes are the proteins inside the cell that are part of are the proteins that promote mitosis, like the receptors for the growth factors and those relay proteins that carry the mitosis signal from the receptor to the enzymes that actually carry out mitosis. And so when those guys mutate to get a gain of function mutation, they're no longer proto-oncogenes, they're called oncogenes, which means cancer genes, because at that point they are forcing the cell to continuously undergo mitosis, and that is a cancer cell. The tumor suppressor genes are just the opposites. Those are the ones that normally act as brakes for the mitosis process. If a mutation causes those ones to lose their activity, so they no longer can act like brakes, that can also lead to cancer. So it really comes down to this: mutations. Here we are. Cancer is caused by mutations in genes related to mitosis, and also in DNA repair genes, because if the cell loses the ability to repair its DNA, then you're going to get more likely to have mutations accumulating in your proto-oncogenes and tumor suppressor genes, and so then you'll end up with cancer. And we talked about these terms of mutagen as anything that increases mutation rates. Radiation is a good example. It causes mutations in DNA. Certain chemicals are mutagens. If you get those inside your body, they get inside your cells and can mutate your DNA. Carcinogens are things that lead to cancer, and almost all carcinogens are mutagens. Cancer is caused by mutations in proto-oncogenes and tumor suppressor genes and DNA repair protein genes. And I think I showed you this, that cancer is a lot more prevalent in the elderly than in young people, simply because the older you are, the more your DNA has had time to be exposed to mutagens. There's natural radiation in the environment around us, and we do things like get X-rays or flying airplanes where a lot of cosmic radiation can hit our DNA. So the longer you live, the more you've been exposed to mutagens, and so the more likely you are to have these mutations that lead to cancer. Here's a graph I pulled off the internet somewhere showing cancer incidence versus age groups. So you can see there's an exponential increase in cancer rates as you go through life. All right, let's see. When I've been showing you these signal transduction pathways for mitosis, I've been showing them like this. There's the receptor for the growth factors, and here's some of these relay proteins that are proto-oncogenes that carry the mitosis signal on downward. There's a tumor suppressor gene, so you finally get the enzymes that actually carry out mitosis, like these S-phase enzymes. This has been This diagram I've been showing you is fairly simplified. There are a lot more proto-oncogenes in the cell that I'm showing you here. The pathway is longer than this. And furthermore, in different types of cells, like liver cells versus bone marrow cells versus pancreas cells versus lung cells, all those cells have slightly different versions of these proteins. And there's a lot of research going on into cancer and mitosis, and they've identified over 100 different proto-oncogenes from all these different cell types and tumor suppressor genes. But that being said, some of these proto-oncogenes and tumor suppressor genes are more likely to be the ones that mutate and lead to cancer than others. And this first one right here, this RAS one, is very commonly mutated in a lot of cancers. When they genetically analyze tumors, in about 25% of tumors, they find a mutated RAS proto-oncogene. There's a tumor suppressor gene called the retinoblastoma gene, and the RB protein is a tumor suppressor protein. It inhibits one of the proto-oncogenes in about, I forget the exact percentage, but in a pretty high percentage of cancers, they find mutations in the retinoblastoma tumor suppressor gene. And at some point I mentioned one called P53, which is also a tumor suppressor gene. There it is. You might remember P53 is a protein that stops the cell cycle at the G2 checkpoint if the DNA has been damaged or in need of repair. And so this protein doesn't repair the DNA itself, but it halts the cell cycle to give the DNA repair enzymes time to repair the DNA. Well, in about half of cancers, this tumor suppressor gene, P53, has a loss of function mutation. And so therefore the cell's DNA repair enzymes just don't have time to repair the DNA before the cell cycle goes through. And so you tend to accumulate more mutations and that leads to cancer. And typically, you need several mutations for a cell to become cancerous. For instance, if RAS were to get a gain of function mutation, I should say an overactive mutation, so it got stuck being too active, that in and of itself wouldn't lead to cancer, because further down the line you're going to have a tumor suppressor gene that's halting the activity of the mitosis pathway. And so you typically need several mutations. You need to lose some tumor suppressor genes by inactivity mutations and simultaneously gain some mutations, some overactive mutations in the proto-oncogenes. And so what I'm saying is when they analyze cancer cells, typically they find mutations, several different mutations that working together lead to the cell becoming cancerous. When they analyze cancers in terms of looking at what sort of things cause the mutations that led to the cancer, there are three major things that are generally responsible for the cancer you find in people. The major one is diet. Here's a pie graph charting the various causes of cancer. And you can see the yellow part here is diet. And simply what it comes down to this is some of the foods we eat have carcinogens in them, chemicals that can mutate our DNA. So when we eat the foods, we get these carcinogens inside us. A good example of that is barbecued meats, or especially overly cooked meats. What happens is, in the heating process of the meats, carcinogens are generated by chemical reactions that go on inside the meat. So these carcinogens were not there in the uncooked meats, but they form when you cook them at high temperatures. And you don't have to know the exact chemicals involved, but if you're curious, I just read about this, so it's fresh on my mind. What they found is that amino acids in the proteins inside the meat undergo chemical reactions with a different type of molecule called creatine, which is also inside the meat. And when these molecules react with each other, they make things called heterocyclic those of you guys in organic chemistry will probably recognize some of these, but it's been a little while since I've had OChem. Heterocyclic amines and polycyclic aromatic hydrocarbons. I'm not sure exactly what those are, but anyway, those are the molecules, carcinogens that are created by amino acids reacting with creatine at high temperature. So I guess the take-home message there is you can lower your chance of cancer if you avoid meats, especially overcooked meats. If you find bits that have blackened edges like that, cut them off before you eat them. Now, at the opposite end of the food scale, what they found is that fruits and vegetables, especially colorful vegetables, actually contain anti-cancer compounds. These are things like lycopenes and beta-carotene and lutins that actually absorb some of the cancerous chemicals that come into our bodies from other sources. And number 2 on the list is smoking. That one's pretty straightforward. The cigarette smoke contains dozens of different carcinogenic chemicals, things like arsenic and benzene, and when you inhale it, these chemicals go right to your lungs. There's a lifetime smoker's lungs right there. You can see how blackened they are from the cigarette smoke versus a non-smoker's more healthy looking pink lungs there. And since the lung tissue is directly exposed to these carcinogens, it's the one that's most likely to get cancer. The people who get lung cancer, 90% of them are smoking, so there's a very definite and real link between cigarette smoke and lung cancer. And the green part of the pie chart there is exposure to sunlight. What it simply comes down to is this, that in the sunlight is a type of radiation called ultraviolet radiation, which directly mutates the nitrogenous bases in DNA. There's a DNA strand, here's some UV light, it just changes the bases in the DNA strand. So since the skin is what's most directly exposed to the UV light, it's what's most likely to where the cancers to develop. And in fact, skin cancer is the most common form of cancer, but luckily it's usually curable if it's caught in very early stages. But if you see dark spots appearing on your skin, especially if they're very darker colors than the normal slightly redder ones that we occasionally see in your skin, that might be something called a malignant melanoma, and we should definitely have it checked out because that's the worst kind of skin cancer. Oh yeah, so what do you do? Well, try to avoid exposure to the sunlight, but if you do go out, slip, slap, slop is the phrase. Slip on a shirt, slap on a hat, slop on some sunscreen. And the other things on this chart, maybe I'll point out real quickly. Viruses, at least some types of viruses, can mutate your DNA. Don't worry if you get a normal cold or flu. That's not the type of thing that's going to cause you to get cancer. But some viruses do mutate DNA, and so I think around 7% of cancers are related to viruses. Occupational factors, people who work with substances that are carcinogenic, like a classic example would be coal miners. I'm sure they have respiratory protection these days, but in the older days, the coal miners would breathe in just a lot of coal dust, and that would cause cancer in the lungs. Asbestos workers used to be exposed to a lot of asbestos fibers and also got lung cancer. Alcohol can lead to cancer. They're not exactly sure what the mechanism is for that one, but when the liver metabolizes alcohol molecules, it changes them into a slightly different molecule called acetaldehyde, which is carcinogenic. And where it says radiation here, they mean things like getting X-rays, or if you spend a lot of time flying in airplanes, there's a lot of cosmic rays up there you get exposed to. And there's a gas called radon gas, which is radioactive, and in some areas of the country can leak out of the ground and get inside people's houses. All right, well, any cancer or general mitosis questions? Okay, well, let us start the genetics chapter. All right, for probably as long as there have been people on the earth, people have wondered about the rules of inheritance and how traits are passed from one generation to the next. And over the history of human beings, there's been a lot of wrong theories about how genetics worked. Like some people thought that traits are always blended with each other. Like if you look at these two people, very dark skinned and very light skinned, and the baby looks somewhere in between. And so a lot of people say, well, traits always just blend to each other as if you had white paint and black paint and mix them together. And in some cases, it does appear that that's the way genetics work. But in other cases, you don't get that. If you have a brown-eyed person and a blue-eyed person having babies, the eyes of the baby are not a mixture of brown and blue. Typically, they would be just brown and not blue at all. And sometimes a trait will skip a generation, where you'll get a trait showing up in generation A, but not in generation B, but it shows up again in generation